Iron is taken up into cells in complex with transferrin and the transferrin receptor, and is then released into the endosomal lumen as the organelle acidifies. Margarida Barroso from Albany Medical College in New York is interested in how the iron is subsequently transferred to mitochondria, where it is needed for a variety of life-sustaining processes.
There was a paper by Sheftel and Ponca in 2007 that suggested that there could be a direct interaction between endosomes and mitochondria, and that would facilitate the transport of iron into mitochondria. And that paper was performed in erythroid cells, which are very interesting for the iron field. Because we do have a very strong background in cell biology and microscopy, we thought that we could leverage that expertise to test the hypothesis that this was a more general situation that would facilitate the transport of iron into mitochondria.
Barroso and colleagues, led by graduate student Anupam Das, used 3D D-Storm super-resolution microscopy to show that transferrin-containing endosomes also contact mitochondria in epithelial MDCK cells. To quantify these interactions, the researchers developed an image analysis protocol.
We didn't want to just go in and identify interactions manually, so we wanted to come up with an unbiased approach, and we took advantage of this algorithm called distance transformation to establish a distance map for mitochondria, and then we could place the endosomes onto that distance map and quantitate the ability of that endosome to be close enough to the mitochondria to interact.
Using this approach, the researchers found that in individual Z-stacks of fixed cells, 5 to 8% of transferrin-containing endosomes were in contact with mitochondria. But do these contacts mediate iron transfer? To address this question, Das et al. Performed time-lapse imaging of MDCK cells stained with the mitochondrial dye RDA.
So that dye emits in the red channel and its signal is quenched in the presence of iron 3. So if the endosome interacts with the mitochondria and upon that interaction we see a quenching in the mitochondria, then that would suggest that the interaction was functional in terms of iron transport. So in the same image we could track an endosome that interacts with the mitochondria and at the same time have multiple mitochondria that didn't interact. And then just follow the intensity of those mitochondria and see the quenching. And at least in the cases that we've tested, we could see a decrease in the intensity of the interacting mitochondria, but not of the non-interacting mitochondria.
Previous studies in erythroid cells have suggested that transferrin-containing endosomes undergo transient kiss-and-run interactions with mitochondria. When Das et al. Followed these interactions by live cell imaging, they saw that the motility of endosomes decreased when they interacted with or kissed mitochondria, which are larger, more static organelles. The researchers then examined the behavior of endosomes in cells expressing a mutant version of transferrin that locks onto iron and doesn't release it into the endosomal lumen.
The endosomes that carry the locked transferrin do not change their behavior in the sense that they continue to interact with the mitochondria. And we do see a reduction of their speed upon interaction with the mitochondria. But when we compare the average speeds, we saw that there was a significant increase in the speed of the endosomes that contain the locked transferrin.
Moreover, blocking iron release with the locked transferrin mutant increased the duration of endosome-mitochondria interactions.
And so there's something happening to the endosome dynamics when you prevent the iron from being released. It could be that the transferrin receptor transferrin complex undergoes conformational changes upon release of iron. So if the iron is not released, those conformational changes may not occur, so that could reflect itself in the cytoplasmic tail of the receptor that would then maybe not interact with motors or whatever that would move the endosomes along the cytoskeleton tracts at larger or slower speeds. The other alternative is that there could be a signaling effect because the redox environment within the lumen of the endosomes changed if the iron is not released. So it is particularly interesting. It opens a lot of research opportunities because it's something we were not expecting.
For now, though, you can learn more about how endosome-mitochondria interactions are modulated by ion release from transferrin in the paper by Das et al., published in the September 26, 2016 issue of the Journal of Cell Biology.